Welcome back to Diabetic Food Files. I'm Dr. G. This is where we explore some science behind the wounds, infections, and then preservations. Forgive me if I'm sounding a little out of it today, but you gotta set your feelings aside and things that you cannot control, like nature. So, today we're going to talk about something that sounds almost like science fiction. If you notice, I've been talking a lot about bacteria, but we're going to talk about kind of like social media for bacteria, and it's called quorum sensing. So it's how bacteria talk to each other. And inside chronic wounds, bacteria are not an isolated organism. They're actually like floating around and create their community. Remember, we talked about the biofilm formation, how it makes it very impenetrable to antibiotics. It makes their antibiotic resistance go up. It's super protective. This extracellular matrix that is sticky, but we're going to talk about how the bacteria can communicate with each other. They actually send signals, they coordinate attacks, they build structured communities, what we call biofilm. And the system that they use to coordinate these different behaviors is called quorum sensing. So I'm going to spell it for you. Quorum sensing. And this is the chemical communication system that allows bacteria to behave almost like a multicellular organism. And when quorum sensing is activated inside a diabetic wound, it can transform a simple infection into a persistent chronic biofilm. So today we're going to break how quorum sensing works, the signaling molecules that the bacteria use, how bacterial communication controls biofilm formation, why diabetic wounds are quite vulnerable to biofilm, and that's why we have to debride, and how future treatments can work to disrupt this bacterial language, okay? What was it in the Bible when they said like they could all speak one languages and then they break us all apart? I mean, forgive me if I don't know the total story by heart, but you know, the bacteria has its own language. So what is quorum sensing? Quorum sensing is a cell-to-cell communication system used by bacteria. And on a side note, medical education, let's make common sense common. And yeah, so each bacterium releases tiny chemical signals into the environment. These signals are called autoinducers. And as the bacteria population increases, the signaling molecules accumulate. And once the concentration reaches something called a critical threshold, the bacteria can detect the signal and they can actually change their own behavior collectively as one, right? So multiple of them can change their behavior due to this signaling that they detect after the population can get to a certain density, like strength in numbers type of thought process, right? So these allow thousands or even millions of bacteria to act as a coordinated group. This definitely has to do with survival mechanisms that they develop. But instead of an individual cell behaving independently, the colony behaves as an organized community. It's like a bacteria mafia, right? So, who discovered bacterial communication? So, quorum sensing was first discovered in a marine bacteria called Vibrophysaceae. And these bacteria live inside certain marine animals and they produce light. But interesting enough, they only glow when the population reaches a certain density. So when enough bacteria is present, the concentration of the signaling molecules trigger a genetic response that activates the light production. And this discovery changed microbiology forever because it showed that bacteria can sense its population density and coordinate their behavior accordingly, which is crazy and amazing, right? So the chemical signal. So different bacteria uses different signaling molecules. One of the most common class of the signaling molecules is acyl. That's how I'm going to pronounce it. ACYL, acyl homosiderin lactones. These molecules are used by many gram-negative bacteria. Gram-positive bacteria often use peptide-based signaling molecules. Another universal signaling molecule that a lot of bacteria uses, like many species, is called an autoinducer 2. Autoinducer 2 allows communication between different bacterial species, which is especially important with microbiology, not micro, polymicrobial infections. Forgive me for saying that. Polymicrobial infections. So the ones that have more than one species of bacteria in that colony, okay? So different bacteria produce different signaling molecules. So a good example is gram-negative bacteria like Pseudomonas aeruginosa, which is one of my favorite bacteria. They use the acyl homosiderin lactones, the AHL, and these signaling molecules diffuse through the tissue and the bacterial membrane. And when the acyl homosiderin lactones, we're just going to abbreviate it and say AHL, reaches the threshold, it causes virulence genes activation, biofilm genes activation, and toxin production increases. Gram-positive bacteria like Staphylococcus aureus and Enterococcus facilis, they use auto-inducing peptides, AIPs, and these peptides activate regulatory pathways controlling adhesion, biofilm formation, and immune evasion. So, what about quorum sensing in a diabetic wound when it comes to diabetic foot vials? So, several major pathogens involved in diabetic foot infections rely heavily on quorum sensing. So, one of the most studied is, like I said, my favorite bacteria is Pseudomonas aeruginosa. This bacteria organism uses multiple quorum sensing systems to control its toxin production, biofilm formation, and it's antibiotic resistant. Another common pathogen is Staphylococcus aureus, which uses a system known as, I'm going to just call it AGR, quorum sensing pathway to help regulate its virulence factors. When the bacteria detects a sufficient population density, they activate the genes that allow them to invade the tissue and evade the immune defense system. So, how does this biofilm form in a diabetic foot ulcer? So, initial step one is attachment. So the bacteria attaches to the tissue or the wound surface using its fimbriae, which are little like tiny, they look like tiny little hairs, okay? Its pillae or adhesion. And the diabetic tissue is vulnerable because of the reduced immunity, the high glucose levels, and the impaired circulation. Step 2, the microcolony formation. So the bacteria multiplies and starts releasing quorum sensing signals, and the small clusters called microcolonies begin forming. Then step 3, they're going to do quorum sensing activation. So once enough bacteria is present, the signaling molecules accumulate, and this activates genes responsible for biofilm matrix production, virulence factor, and antibiotic resistant mechanism. And step 4, the biofilm matrix production. So bacteria produces a protective polymer matrix called extracellular polymeric substance, so EPS. And in this EPS, this protective polymer matrix, it contains polysaccharides, proteins, extracellular DNA, and lipids. It forms a gel-like shield around the bacteria. Remember, it actually increases its antibiotic resistance. It makes it harder to kill this bacteria off because it has this protective shield. So, like I said, one of the most important things for quorum sensing controls is biofilm formation. So, when the bacteria detects that there are enough of their neighbors present, they begin producing the extracellular matrix that forms the biofilm. And these together form the extracellular polymetric polymeric, forgive me, substance that protects the bacterial community. And once the matrix forms, the antibiotics that we need to kill the bacteria off struggle to penetrate the colony, and the bacteria colony can just thrive. And the immune systems have quite a difficult time reaching the bacteria once there is a biofilm formation protecting them. So, why are biofilms so dangerous when it comes to diabetic foot ulcers? We already know diabetic foot ulcers, their immune system is already compromised because of this disease. And biofilm formation dramatically changes the bacterial behavior. They feel a little cocky, right? Because they're protected now. So, inside this biofilm bacteria, the bacteria has up to 1,000 times more antibiotic resistance. So, like I said, the antibiotics struggle to penetrate the, extracellular polymeric system matrix, and also the bacteria inside the biofilm enter something called a metabolic dormancy, which makes antibiotics less effective. What also happens when they form this biofilm? It gets hidden from the immune system. The immune system cells, such as neutrophils, cannot penetrate the biofilm. So, this is why it leads to chronic inflammation, which is the stage that a lot of diabetic foot ulcers get stuck in, persistent infection, and tissue destruction. Now, producers of virulence factors like quorum sensing activates toxin production. So, we're going to use our favorite bacteria, Pseudomonas aeruginosa, as an example. When it comes to quorum sensing, the toxins that it activates for Pseudomonas aeruginosa is elastase, protease, and piocyanin. 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 And these destroy tissue and delay healing. So, why would they ideally want to make a biofilm in a diabetic foot ulcer? So, diabetic wounds create a perfect bacterial environment. They see it, they want to thrive in it. So, the high glucose level. Remember, sugar is quite amazing fuel for bacteria metabolism. Okay, so they had as much glucose as they like. So, that's another reason to control your blood sugar so well, because you don't want to add fuel to the fire, right? If your blood sugar is so high, it's actually aiding the bacteria fuel. Okay, it's helping it metabolize, it's helping it multiply. You're giving it this fuel. You're not suffocating it, you're not starving the enemy, right? But we're not saying starve yourself, but you have to control your blood sugar. Remember how dysfunctional the immune system is with a high blood sugar? Next is hypoxia. We all know that a lot of diabetics deal with poor circulation issues. And what does poor circulation result in? Reduction in oxygen. So low oxygen environments actually favor biofilm bacteria. Next, like we said, the impaired immune response. So diabetes, like we said, causes dysfunction on the neutrophils, the macrophages, and the cytokine signaling. This allows the bacterial colonies to continue to expand if all of these are dysfunctional. Now, why would a diabetic ulcer or wound trigger quorum sensing? Like we said, it's the perfect environment for bacterial communications. The several factors that can help with that is the poor blood flow, the neuropathy, the elevated glucose levels, the impaired immune function, and these conditions allow the bacteria to multiply without quickly being eliminated, okay? And as the population grows, the signaling molecules accumulate, and eventually the quorum sensing threshold is reached, and the bacteria switches into biofilm more and be like, make more and more. So like when you see the biofilm, they're like, make more and more, it's all I think about when I hear about stuff like this. I think of Game of Thrones. I love me some Game of Thrones, House of Dragons, all that jazz. But it's all about that, okay? So, what does the evidence say from research? So, studies show about 60 to 80% of chronic wounds contain biofilms. And in diabetic foot ulcers specifically, biofilms are commonly composed of polymicrobial communities. And what usually affects diabetic foot ulcers are usually polymicrobial, okay? So let's say they might have Staphylococcus aureus in it, Pseudomonas aeruginosa, and Enterococcus facilis, okay? But these organisms will be able to cooperate together using interspecies quorum sensing, okay? So quorum sensing doesn't just build biofilm, but like we said, it also controls the release of toxins. Like I said, Pseudomonas aeruginosa produces destructive enzymes that break down tissue. the enzymes, like we mentioned, hemolysin, elastinase, protease, the bacteria does not produce these toxins immediately. They wait till they have strength in numbers, till there are enough bacteria present to overwhelm the host defense system. So, see, they don't just start off as one because they know the moment that there's just one, the person might notice antibiotics immediate, wiped out. They literally sit and wait till they accumulate enough where it's harder to get rid of them, right? Keep on saying strength in numbers, okay? So once that quorum threshold is reached, the toxin production increases dramatically. So, quorum sensing regulates genes involved in antibiotic resistance as well. So, inside the biofilm, bacteria often enters something called a slow metabolic state, and many antibiotics targets rapidly dividing cells. So, they kind of overlook the slow growing bacteria. So the slow growing bacteria are less vulnerable compared to the rapidly growing bacteria. So in addition to the biofilm communities containing the persister cells, these cells are highly tolerant to antibiotics and can repopulate the biofilm after treatment. So that's why biofilm comes back so quickly, and that's why debridement is such a big deal. So why does debridement work? Because it literally is the physical disruption of the biofilm architecture. We're not just wiping it with water. Remember, it's sticky. It's polymeric extracellular matrix, okay? So it's gooey a little bit, it's sticky. So, ever saw like sticky glucose and like wiping it with sugar? It's like sugar, but wiping it with water, it doesn't do much, okay? You actually need to like get a knife and scrape it like molasses. It's like molasses, okay? So once the biofilm is broken, the bacteria becomes planktonic, and the antibiotics actually become effective for it to penetrate, and the immune system can actually attack. But however, the biofilm can reform quite quickly. Biofilm actually forms within 24 to 72 hours, okay? And that's why repeated debridement is very critical. So patients can understand that we're not just trying to cut you, we're trying to remove the biofilm that this bacteria is trying to gain resistance and take over the wound itself for itself, right? They're trying to literally Columbus the wound. They're like, we found it, we're taking over. So, because quorum sensing plays such an important role in infection, researchers are trying to develop something called quorum sensing inhibitors. And these compounds actually block the chemical signals bacteria uses to communicate. So, if the signal is blocked, the bacteria cannot coordinate biofilm formation. They cannot synthesize toxin production, and the infection becomes quite easier to treat. And there are actually some natural compounds that show that they can inhibit quorum sensing in bacteria, including curcumin, and then some compounds that are found in black seed oil. So what are some of the future therapies that they're trying to combine with traditional antibiotics to help with quorum sensing inhibitors? So things that block bacterial communication. So compounds, like I said, examples are going to be, they're really studying plant-derived molecules and enzymes that disrupt the biofilm, bacteriophage therapy, so viruses that actually target bacteria and penetrate the biofilm. So they literally are using viruses to target bacteria. People always people try to interchange bacteria and viruses, but they're two completely different things. A virus cannot survive without a host. Like, it needs a host. Bacteria can survive and thrive, and they could do it on their own, right? But viruses and bacteria are completely different things, okay? But that's why they're going to use a virus to target the bacteria, okay? And this strategy could prevent biofilm formation, reduce virulence, make bacteria more vulnerable to the antibiotics. And like I said, it's going to be bacteriophage therapy or enzyme-based biofilm disruptors. And then also something that's coming about is this called nanoparticles that target the bacterial communication pathways. So the goal for these are not to kill the bacteria, but actually silence their communication so they won't have strength in numbers. So in every chronic wound bacteria is not just surviving. It's actually communicating, it's organizing, they're getting smarter every day. They're building a protective structure and coordinating their behavior through quorum sensing. So, understanding this hidden language that bacteria use to protect itself explains why some infections are so persistent. They actually learn your body. So, knowing this actually opens the doors to new therapies that may help clinicians one day break down better for bacterial biofilms formation effectively. So, in a diabetic foot ulcer, bacteria aren't just infecting a wound, they're talking to each other. They're like little, like I said, it's like social media. That's why social media is so powerful these days, right? Because you can get anywhere, reach to anyone, any time of the day, right? So that's quorum sensing for bacteria. They coordinate, they attack, they build their biofilms to turn a wound into a microscopic fortress. So thank you for listening to Diabetic Foot Files, where we explore the science behind wounds. And remember, sometimes the most dangerous conversations are the ones that are happening at a microscopic level. So this is Diabetic Foot Files. I'm Dr. G, and take care of your feet because they take care of you.